Hello all, welcome back to the BioRocks channel. In this video today we'll be learning about the development of the female gametophyte in the angiosperms and remember children, the female gametophyte actually is also referred as embryosac or the mega gametophyte. Okay, this develops within the ovule. Okay, so that we are going to learn today, that how an embryosac is going to develop. That is your female gametophyte of the angiosperms. As you can see it on your screen as the carpel or the gynoecium, where you can see the ovules embedded, and this is going to have the archisporial cells, where one is going to differentiate as a megaspore mother cell, which undergoes a series of divisions, and at last is going to develop an embryo sac, which is being called as a female gametophyte. Okay, children. So what I'll do is I'll go into the details of this. I'll make it sure that it will be easy for you all to understand, and that is going to help you in scoring your marks of the boards as well as the NEET. Okay, so I'm entering into the details of this. The first thing that you have to concentrate is an ovule is going to have the hypodermal cell that differentiates into the archisporial cells. Okay, so first what we are seeing is we are going to look at the ovule. Okay, so if you look at the ovule, for instance, if the funicle, this is going to be the ovule. Okay, I'm not talking about the embryo sac. Okay, I'm talking only about the ovule. So the ovule is going to have some cells, and these cells are actually the ones which are being called as hypodermal cells. Hypodermal cells. Amongst these hypodermal cells, now what happens is the cells are going to, now these hypodermal cells are going to differentiate. They are going to differentiate into the cells which are being called as archisporial cells. Archisporial cells. So now amongst these archisporial cells, children, these all cells are going to be collectively called as what? Now they'll be called as nucellus. As you know that the ovule is going to contain the tissue, right? Ovule, if you're going to look at an ovule, the ovule is going to embed the tissue, which is being called as what? We call it as nucellus. So who is making the nucellus? These are the archisporial cells. Do you understand now? So all the hypodermal cells have differentiated into the archisporial cells, and these archisporial cells are actually, they are diploid. Okay, they are diploid in nature. All these diploid cells, the archisporial cells, are going to collectively be called as what? We're going to call it as a nucellus. We are going to call it as. Now, what happens is at the maturity. So now at maturity, what happens is one of the cell, okay, at the maturity, one cell amongst these archisporial cells is going to differentiate as megaspore mother cell. One cell is differentiating now. Okay, this is differentiating into a megaspore mother cell. A megaspore mother cell. Okay, children. So the megaspore mother cell is also diploid, right? This is diploid. But now this megaspore mother cell will be undergoing meiosis. So it is undergoing meiosis. So because of the meiosis process, now what happens is this is going to form four haploid haploid. Let me not confuse you because this will be something else, the notation. So four number of haploid megaspores it is going to form. Okay, the cells, the nucleus is being called as megaspores now, and these are going to be haploid in nature. Okay, and these can also be called as tetrad. Tetrad. They are being arranged one above the other, so we call it as linear tetrad. Okay, now The formation of these megaspores from the megaspore mother cell. See children, this process. The megaspore mother cell forming these linear 4 megaspores. This whole process can be defined as megasporogenesis. Megasporogenesis. Important for the short questions where you are being asked by the uh, definition here. So, what is megasporogenesis? A diploid megaspore mother cell undergoing meiosis and producing haploid 4 linear tetrad megaspores is being called as megasporogenesis. Did you understand? So, ovule has a hypodermal cells which differentiate as archisporial cells. Amongst these, one of the cell forms megaspore mother cell, which undergoes meiosis to produce a megaspores, which are haploid in nature. These are being called as megaspore. This process called as megasporogenesis. Okay, children. So now, amongst these 4, which are being arranged linearly, three of them are going to degenerate. So these 3 are going to get degenerate. Okay. And now what happens is the one at the base is going to become functional. So let me color the functional for you. So this is going to be the functional one. It's a functional megaspore. The 3 are getting degenerated. Okay. Now what happens? It is the first cell of the female gametophyte. Who the tetrad, the functional megaspore, is the first cell. This is the first cell of female gametophyte. Okay, children. Who is the first cell of the female gametophyte? The functional megaspore. Okay. Now, this megaspore, this functional megaspore, now what it does is it undergoes mitosis. Okay. It undergoes mitosis. So, how many nuclei will be produced? 2, right? Because it will undergo what? Mitosis process. It will produce 2 of the nuclei now. Okay. Now what happens is these nuclei will be migrating to the opposite ends. One to the top, one to the bottom. Okay. They are being moved apart. Now what happens is these are producing 2 nuclei and these nuclei are moving to the opposite poles of the megaspore, and at each pole the nucleus divides twice, children. This will be the first division. Okay. Now, first division, how many are produced? These many. And next it is going to produce how many? The second mitotic division, because it is going to undergo the nucleus will divide twice. Okay, first it has divided, and the second time it is going to divide, producing 4 nuclei at each pole, right? Here also the second phase of division. This is going to result in 1 2 3 4, 1 2 3 4. Okay, this is a second, this is a first mitotic divisions. Okay, so it has undergone 2 divisions, children. Again, so the ovule has a hypodermal cells where they differentiate to form archisporial cells, where the one cell is going to differentiate to form a megaspore mother cell, which undergoes meiosis to produce 4 megaspores. Amongst them, 3 will be degenerating and 1 will become functional. That is the first cell of the gametophyte. And that cell is going to undergo what? The mitotic cell division to produce 2 nuclei. Okay. Now what is happening? These nuclei are migrating to the opposite poles. Okay. They are moving to the opposite poles here and here. And later what has happened after moving to the opposite poles now? At each pole the nucleus is dividing twice. Okay. The first and the second mitotic divisions. And producing the 4 nuclei at each pole. Here also 4. Here also 4. Now the functional megaspore enlarge gradually. Okay. And these becomes a 8 nucleated embryosac. Now, this is very important to understand that the one which is present here, which one? The functional megaspore. So the functional megaspore, let me draw it with another color. So this is the functional megaspore. Okay, the functional megaspore having the 2 nuclei, right? They move to the opposite poles here. This is your megaspore, where there is a second set of mitotic division. And now the megaspore is comprising of what? 4 nuclei at each pole. Is it clear for you now? So the functional megaspore has got how many nuclei first? It has got one nuclei which has divided by mitosis, produces two, moves to the poles, and they are undergoing two phases of mitotic divisions, and later they are going to form the four functional, that is four of the nuclei at the each of the pole. Okay. Now children, remember that the four totally, now we are going to see that the functional megaspore is having how many nuclei? This is your functional megaspore, right? This is our functional megaspore. This functional megaspore is having how many nuclei now? 8 nuclei. Okay, so what do you call this as? 8 nucleated embryo sac. This is your 8 nucleated embryo sac. Okay, so the two nuclei, one from the each pole, will migrate to the center now. So let me take a next page. So I have got a embryo sac with 4 nuclei here and a 4 nuclei here, right? Okay, now what is happening? The two nuclei. So if this is my embryo sac, two of the nuclei are moving from each pole to the center, and they are fusing to form a diploid nucleus. Okay, they are fusing. Now this structure which is going to form here is going to be a diploid secondary nucleus. Okay, this is your secondary nucleus. Next, the three nuclei at the chelazal end will be here. So these will be your 3 cells at the chelazal end. These will be called as what, children? They will be called as antipodals. Okay, so antipodals are towards which side? They are towards the chelazal end, from where your integuments are arising. Okay, and you can even see that there are 3 of the cells, right? You can see even 3 of them here. Okay, so you can see that the amongst these 3, you can see that they are the central cell here will be called as the egg cell or the egg apparatus, and the other 2 will be called as synergids. Okay, the peripheral ones are being called as synergids, and the central one is being called as egg cell, or we call it as egg apparatus. Okay, children. Now the embryo sac is having how many cells? One, that is 2 synergids, 1 egg apparatus, 1 secondary nucleus, and 3 antipodals. So togetherly how many? 7 celled. Right? 7 cell. But how many nuclei are there? How many nuclei? 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleated. The magic is here in the secondary nucleus where the two nuclei fused, right? So this is going to be such type of embryo sac. Okay, 7 celled, 8 nucleated embryo sac is called as polygonum type. This has been called as a polygonum type embryo sac. Okay. And the development of the female gametophyte in angiosperms is completely endosporic, children. That is, within the megaspore and monosporic, right? Because the female gametophyte is developing inside the megaspore. So what do you call it as? Endosporic. Let me write this for you. The development of the female gametophyte is endosporic, right? Development of female gametophyte is which type? It's going to be endosporic. Why endosporic? Because it is developing inside the megaspore. Right? It is developing inside the megaspore. And it is monosporic also. It is monosporic. Why monosporic? Because it has developed from a single megaspore. Did you understand? I'll write this for you now. So it is endosporic because it develops within megaspore. Who? The embryosac. Right? Who is developing? Embryosac is developing within the megaspore. Why is it monosporic? Because it is developing from a single megaspore. So we call the embryo sac or the female gametophyte in angiosperms as a polygonum type, endosporic, monosporic. Do you understand these now? These are important. But remember, there are even few angiosperms where it can be bisporic and tetrasporic also, children. That is also important for you to understand. But you can see that in synergids, there are special finger-like folds, and those will be called as filiform apparatus. They will enable the let in of the pollen tube. Filiform apparatus are found in the synergids only. This is also important for you. Now let me show you how this process is being done. I'll show you one of the slides that will help you in this, understanding it more far better. Okay. So, yeah, children. So this is the one that you can see on the screen. As you can see that inside the ovule, the archisporial cells, that is the nucellus, one of the cell is differentiating as a megaspore mother cell, which undergoes meiotic cell division process, produces 4 haploid daughter cells, and 3 of them are going to degenerate. One is going to be left, which will be a functional megaspore. And in the functional megaspore, this is going to divide. The nucleus divides by mitosis, produces 2 of the nuclei. So this is your megaspore. Okay, and these are the 2 nuclei. And now these will undergo 2 phases of mitosis. The first stage, the second stage, 2 times it undergoes mitosis and produces 4 of the nuclei at the different poles. Okay, and this is going to be the mega gametophyte. Who is a mega gametophyte? The embryosac, right? So now you can see that amongst these 3, the 2 of them from each of the poles are traveling and forming a secondary nucleus, and one towards uh, the micropylar end will be forming the synergids and the egg cell, and the opposite end ones towards the chelazal end will be forming the antipodals, right? So this is going to be the procedure. And let me show you another slide where we have again the, not this, let me go to the other slide that I have got for you to show you the development of the female gametophyte here. So children, you can see that it was one megaspore mother cell from where the embryo sac is developing. So we call it as monosporic. And since inside the megaspore only all these, the embryo sac develops, we call this as the endosporic also. Okay. Now the 3 of them are degenerating because the megaspore mother cell undergoes meiosis, produces 4 haploid tetrads, and 3 of them are going to disintegrate, degenerate. One is left over, which is a megaspore or the embryo sac, which will undergo the mitosis, produce 2 daughter cells, where they undergo 2 stages of mitosis, again producing 4 of the cells at each pole, where 3 of them will be the antipodals, 2 of them will be the synergids with 1 egg cell, 2 of the nuclei will fuse to form the secondary nucleus, and we get the mature embryo sac like this. So this is the development of embryo sac, which we call as female gametophyte in the angiosperms. I hope you all have understood the procedure. May this video might have helped you in understanding it far better. If you like this video, please do hit the like button. Try to share and subscribe my channel. Please do comment in the comment section how you like this video. Thank you all. Thank you.